Let's say you're in a room with your nephew, D and A. D just got done playing with his Legos. Now he built a certain pattern of Legos that he really likes, but it's scattered in a room of Legos and he just can't find what he's looking for. Luckily, there's only 4 colors: light blue, red, pink, and green. But D is full of imagination though, so he renamed the colors aqua, tomato, carnation, and, well, green. Interestingly, the aqua piece always fit perfectly with the tomato piece, and the carnation piece always fit perfectly with the green piece. Now D is asking you to build a new pattern just like the one he built, but you have no idea what it looks like, so you can't help him. Luckily, his friend just arrived and wants to play. His name is Tak. D asked Tak if he remembered exactly what pattern of Legos D made. Unfortunately, Tak doesn't remember either, but he remembers the first few in the pattern. He called it the primer. So he looked everywhere, combing the entire room of Legos to find the pattern he was looking for. Once he found it, he attached the aquas with the tomatoes and the carnations with the greens, and he continued to add them together until the whole pattern was made. He then pulled the 2 long strands apart and then again added the A's to the T's and the C's with the G's until they became 4 strands. He continued to do this until there were enough of the same strands present that you could actually see the pattern in this big sea of Legos. You thanked Tak for his help and he went back to his house over by the hot springs. Now, when biochemists want to identify and study exact nucleic acid sequences, they too need to make many copies of the same sequence, just like you needed to make many copies of the Lego pattern to see what it actually was. Biochemists use a strategy very similar to what we did here. It was invented in 1984 by American biochemist Kary Mullis, and in this video we're going to be talking about this technique. This is Joel and Matt talking to you today about the polymerase chain reaction.
Our presentation today will cover basic theory and principles behind PCR, and we'll also discuss with you the history of background of the procedure. We'll talk about the materials and as well as the procedural steps that are used in PCR, and then we'll discuss technological advancements in PCR along with applications and limitations of its uses. Like any new technology, PCR's advent came about as a solution to a problem. The problem was that DNA samples were far too minuscule to produce any meaningful analytics. This was made even more difficult by the sheer size of genomes. As a solution to the problem, PCR is able to take a small sample size, we're talking a few or even a single strand of DNA, or in some cases RNA, and produce 1,000 to 1,000,000 copies for further analysis. This technique was developed in 1983 by Dr. Kary Mullis, who was working at the Cetus Corporation at the time. Dr. Mullis was tasked with producing oligonucleotides for the research team there, and his initial plan was to use Sanger sequencing in conjunction with DNA polymerase and nucleotides. But as I mentioned earlier, the sheer size of genomes made this task way more arduous and difficult than it should be. He developed PCR with the intention of only synthesizing the sequence of interest. Ultimately, Mullis received a Nobel Prize for his work in developing the procedure.
So let's go over the list of materials needed to perform PCR. First, you obviously need the gene of interest, taken from whatever sample you're testing. Next, you'll need a primer. The primer must be specific to flank the segment of interest. They require engineering, and they require high guanine cytosine content, and we'll talk about why in a bit. And finally, the primer needs to be heat resistant. As you'll see later on going through the steps, the DNA strands are subjected to cycles of heat to break apart the double strands. If the primer itself is unstable at high temperatures, it too will denature in the process. Next, you'll need the actual DNA polymerase itself, which, if you recall from general biology, is your main protein responsible for taking free nucleotides and attaching proper complementary nucleotides to the new growing nucleotide strand. E. coli is often used to study mechanisms at the molecular level due to its abundance and ease of use, but unfortunately its polymerase degrades at high heat. So biochemists saw this and recognized that in order to replicate a DNA strand that has been recently separated by high temperatures, the DNA polymerase itself must be resistant to high temperatures. So instead, biochemists use isolated DNA polymerases from Thermus aquaticus, a microorganism that is an extremophile. More specifically, it's a thermophile that can survive in temperatures of up to 80 degrees Celsius and is found near hot springs. Using Taq polymerase, we can subject the DNA to successive heat shocks to break apart the double-stranded helix, while still having an intact polymerase to synthesize the new strands. Taq polymerase will need to add nucleotides to the growing strand, so an excess of free deoxynucleotide triphosphates, or DNTPs, will need to be added to the solution. As you might expect, the 4 DNTPs to be added are DATP, DCTP, DGTP, and DTTP for adenine, cytosine, guanine, and thymine. These are essentially your building blocks. So now let's go over the actual steps of PCR. There are 5 major steps: DNA isolation, denaturation, annealing, extension, and final cooling. The first step is to isolate the DNA. It can be done using organic extraction. This involves lysing the cells and using denaturing solvents to break down cellular components. Then the nucleic acids are extracted separately. Another method is spin column chromatography, where the solution is placed in a spin column for rapid purification. These columns allow convenient purification of DNA and PCR products greater than 200 base pairs directly from reaction mixtures. Spin columns eliminate the need for matrix binding and wash buffer steps, gravity columns, dialysis membranes, phenol extraction, and alcohol precipitation, all of which are time-consuming to isolate PCR products. The second step is denaturation through heat. The double-stranded DNA is subjected to high temperatures which break the hydrogen bonds holding the double strand together, much in the same way heat breaks double bonds in proteins to dismantle the secondary and tertiary structure. The target temperature for denaturation is between 92 and 95 degrees Celsius. More hydrogen bonds equates to higher temperatures required. The third step in the process is annealing. Now, during annealing, the temperature is decreased down to 50 to 56 degrees Celsius. This is the optimal temperature for hydrogen bond formation. In this step, 2 primers anneal to the DNA template. This signals the beginning of DNA extension by Taq polymerase. Because the DNA is now separated into 2 strands, both the forward and reverse primer flank each side of the region of interest. Shown here are the 2 strands, both the sense and the antisense strand in anti-parallel configuration. One strand goes 5' to 3', and the other goes 5' to 3' in the other direction. Now, primer design is crucial. It must be specific and complementary to the target strand, ideally with a high guanine cytosine content. This is because of the stickier nature of the guanine cytosine bond, which is ideal when trying to apply the primer to the DNA template strand. Guanine cytosine content is an important consideration because of the nature of the hydrogen bonds between them. Shown here is a thymine paired with an adenine on top, with a guanine and cytosine on the bottom. As you can see, there are only 2 hydrogen bonds between adenine and thymine, but 3 hydrogen bonds between guanine and cytosine. Because of this, higher GC content confers greater stability in the double-stranded structure. The fourth phase is synthesis or extension. During this phase, temperature is increased to roughly 72 degrees Celsius, which is the optimal temperature for Taq polymerase function. With the primers annealed, Taq polymerase comes in and adds nucleotides complementary to the template strand. The mechanism of phosphodiester bond creation won't be covered in detail in this video, but just be aware that like all DNA polymerases, the Taq polymerase here creates covalent linkages between each nucleotide. Once the strand is complete, you are left with 2 double strands of DNA rather than 1. At this point, the process can just simply be repeated. Here you see the thermocycler temperature increase just like in step 2. The 2 double strands become 4 single strands. Step 3 is repeated, the temperature is decreased to 50 degrees to allow for primer annealing. And step 4 is repeated by increasing the temperature to 72 degrees, once again allowing Taq polymerase to extend the primers and leave you with 4 double-stranded DNA segments instead of just 2. Because Taq polymerase is able to endure thermocycling, the entire process can be automated up to 20 to 40 cycles or until sufficient DNA is present for analysis. And finally, after the final cycle, a final cooling down to around 40 degrees Celsius allows complementary DNA strands to associate and prevents degradation.
Next, we'll talk about some technological advancements in PCR. One major technological advancement was the development of quantitative PCR, also known as real-time PCR. This technique uses fluorescent signals to quantify the concentration of the nucleic acid sample. Fluorescence is introduced to the nucleic acid sample using one of two methods. Fluorescent intercalating dyes can be added to the solution and highlight the sample by sitting in the spaces between the base pairs. A second method of introducing fluorescence is the use of fluorescently tagged nucleotides. A major benefit of real-time PCR is not only that it quantifies the data, but it does so in the midst of the amplification process. This allows researchers to build concentration models quickly and use fewer cycles. A second major development is reverse transcription PCR that uses reverse transcriptase enzyme to convert RNA back to DNA for the same amplification process. This process is commonly paired with qPCR. Next, we'll talk about some modern usage for PCR. In the medical field, PCR is used for paternal testing, prenatal testing, as well as cancer and other disease diagnostics. Target sequences are amplified and compared to either the father's DNA in the case of paternity testing or sequences known to be associated with diseases. PCR can often be exploited for prenatal testing to diagnose potential aneuploidies present in fetal tissue. The laboratory settings, the same PCR process is commonly used for cloning, mutation, genotyping, and forensics purposes. Like any technology, limitations arise. With PCR, limitations include the fidelity or error rate of the DNA polymerase. In this case, Taq polymerase has an error rate of approximately 1 in every 3,500 base pairs. Nonspecific binding can also occur. Misaligned primers will affect the sequence that is produced. Lastly, we encounter difficulty in engineering the primers that are used, which requires extensive knowledge of the template sequence in the first place. Thank you all for watching our video. We hope you enjoyed it. Dr. Wassenberg will have some questions for you to answer.